So welcome everybody. We are making sure that everybody is connected. Sorry for the delay. It's good to see old friends and new friends here. So that's wonderful. So I want to welcome you on this week's or this month's meet and greet. Ioannis Kakadiaris, I'm a professor of computer science at the University of Houston, and also I'm the chair of this new cluster. And I want to thank everybody for taking the time to present and to attend this virtual meet and greet. Today we have 4 volunteers, and we have even a volunteer that has not joined the university yet, but that's wonderful. We are looking forward. So I'll pass the microphone to Anand, who is responsible for the first session. Anand?
Great, thanks, Yanni. So welcome everyone. My name is Ananta Napragada. I'm a professor in radiology at Baylor College of Medicine and Texas Children's Hospital. And I have the privilege of being one of the co-chairs of this cluster, along with Yanni and with Devika Subramaniam, who will follow up with the next session of 2 talks. So the first short talk today is by my colleague from Baylor, Dr. Rainer Lanz, who is among other things, an expert on the progesterone receptor. And I look forward to hearing what you have to say in the context of this cluster, Dr. Lanz.
Well, thank you. I don't know what I can say in 3 minutes, though. Okay, I mean, you see my name here. I'm not reading what you can read. You see an actual photo. I took this yesterday here in my office, so everything is up to date. My research interest, I added 2 pictures. I don't have control of the slides, so I have to point it. So I'm a passionate photographer, so I illustrate the 2 main things that I'm interested in with these 2 figures here. One shows 3 colored filters that I overlay, and this stands for actually integration, data integration. And the simple message here is, you see more, or you see things that you don't see if you would analyze the data individually. The second picture to the right shows, it's a book cover, by the way, that shows 2 data sets and illustrates how one way I analyze data by looking at the projections. If you look at the projections here, you see exactly that there is an intrinsic dependency between these 2 data sets. You all know machine learning. We have more complex projections than the linear projections as illustrated here. But that should illustrate in, that I like to do data analysis, and I use integration and modeling for the analysis. I do a lot of consulting, although this is committing almost professional suicide these days, because you are evaluated only on efforts and how much money you bring in. But I do a significant amount of consulting. I help people out left and right, okay? And I'm very interested in concepts more than actual data. I will get back to that soon. Unique resources. I didn't know what you really want here. Maybe computer power or something like that. I don't think a computer is anything unique. So people say I have a good brain. I have a master's in biochemistry. I have a PhD in molecular biology. I have 2 years math and physics at the ETH in Zurich. I'm versatile, very versatile. So I have high versatility, they say. And I'm 40 years in this field, so I have some experience, I guess, right? What I can offer, though, is actually my classes. So I mentioned here 1, explorative data analysis. These are 24 hours, term 3 classes here at Baylor. And I allow people to sit in. So you're welcome to attend those. This will be distance learning this time around, I guess, right? So the next slide. sorry, I have no control, okay? What can you contribute to the cluster? Here's go, the curse again, okay? But I'm trained on gene expression, especially nuclear hormone receptors and co-regulators. As mentioned, one of the nuclear receptors is progesterone receptor, but I started out with GR and do a lot of ER, and these are all involved in cancer. And I was also very early on with the co-regulators. These are transcriptional co-regulators for mainly the nuclear hormone receptors. As I mentioned already, I do applied predictive modeling and consulting and that, so maybe that could be a contribution to the cluster. And since 2002, I promote databases in general, especially related database management system. I still see people going up and down with their fingers on up and down the Excel sheets and things like that. I think this should go away. And a lot what I do is I use relational database systems to do the data analysis. What can I contribute? I mentioned here cold deck imputation, and I have two figures here that may illustrate what I mean with that. This is a Cell paper that we had a couple of years ago. We analyzed actually how nature put proteins together, so complex somics if you want. The left-hand side shows in the heat map that no matter how many experiments you analyze, you have three components that are always showing up, while the rest is very variable. So we defined a term called MEMOs, minimal endogenous modules, that then can combine with other proteins to core complexes. At that time, we called it unicores, which then can form complex interactions and are part of pathways and things like that. So a memo, if you know that you have a memo of 3 ABC, for instance, and you take only one, we should assume that B and C should also be in the found set, although we did not detect it. So this is what I mean by cold deck imputation. And the next slide, then don't change slide now, but the next slide, the next slide will show more about that, what I mean by that, okay? So what do I expect from the cluster? I don't even know what the cluster is. So what can I say what I expect from the cluster, okay? But I would be interested in deep generative models, graph neural networks, and a little bit about soft computing. Why? Because the next two illustrations show, or maybe one illustration if combined, shows how I perceive the data space that we have to deal in our professions, okay? So we have the horizontals platform illustrate different data sets coming from different platforms. We have sequencing data, we have clinical data, we have metabolites, et cetera, okay? But while AI already is applied horizontally in all these data sets, I think we do need more integration. Why? Because yes, you can have feed-forward or feedback loops on gene expression, but gene expression is not the end point of the physiology. You have to go through protein. Proteins go form complexes. Complexes are part of some higher order events. And so this illustrates that I promote integration through different platforms. And that is the data space determined by how many features you have, how many samples, and how you go vertically through the space. This gives then the prediction accuracy of what AI, I think, could do. RFA, I mentioned here too, I'm not affiliated or not part of any of these two, but for the decoding and modulating neural circuit activity linked to behavior, I consulted a group here at Baylor that analyzed actually quite extensive data from people that have committed or tried to commit suicide or expressed to kill themselves. And they were analyzed with brain scan, and I composed or I advised on the data modeling to analyze this data. And the second one I mentioned here is just because this is training. So we are always involved in cancer, and the data sets that we are analyzing here is quite often in cancer. The last slide, I think I'm over 3 minutes already. I thought I share with you what I think AI can do for us, for biomedical science in general. As I indicated already, the horizontal red arrows indicate that AI is, I hate AI, by the way, as a term, okay? I prefer more specific like machine learning, deep learning, or reinforcement learning or something like that. But it is applied already in within the data sets itself, you know. But as I alluded already, we need more vertical approaches. We need more cold deck imputation, as I indicated in the previous slide. We need more soft computing. And here I show you a very simple screenshot of a logistic regression. We analyzed prostate cancer, gene expression data sets on 300 and some men. And the clinical manifestation is very simple. It's Boolean. You're either healthy or you're sick, right? These are the blue circles that you see at 0 or at 1, you know. But the data does not put the patient there. The data puts the patient where you have the green or the red crosses there, you know. And the Bayesian decision boundary is 0.5. If you are slightly above 0.5 or slightly below 0.5 from that data set, you are the same. Although clinically you are either sick or either healthy, okay? So we need more probabilistic models, such as deep generative models like Boltzmann machine that are structured probabilistic models. Along the same line, I think AI will go into more deep learning or reinforcement learning, but not on data as it's done right now. I hope on relationships. This will bring us then to graph neural networks. That's I should have put that term there actually, okay? And lastly, because I'm passionate for getting what I know to students, we need a little bit more for knowledge type of education. So we need to go back 40 years probably and teach the students to understand what they're doing and not sign up for this kit mentality. But that opens a whole other can of worms, I guess, and I can discuss an hour on that term alone, okay? So I don't know whether I introduced myself.
Well, this was...
I had only 3 slides, so there is no 4th slide, sorry.
This is great. We're obviously not strictly enforcing the time because we only have 4 speakers, and we might as well get into the depths of the conversation better.
Yeah, you can.
Thank you very much for that great talk. Any questions from the listeners?
Just a comment.
Yep.
I really appreciate your approach to education. It's going to be an uphill climb, so I'd love to meet with you to see how we can get, go back 40 years. That sounds about right. And also, I agree with you that the AI has been done on each layer, but not across. And I think part of it is really that we don't have data sets on the same individuals that go across the stack vertically. So one of the biggest problems, I'm a computer scientist by training, and it's been very hard to find coordinated data sets that cross the entire stack. So if you have ideas about that, I'd love how to get it. Then maybe, that's something the cluster can try to get. And maybe you tell us how we can go about doing that. So that would be great.
Yeah, if you want, my thoughts on this is there are a lot of entities, university, hospital, big hospital that actually aim for 100,000, 1 million genomes, or even 10 million genomes now, right? And Baylor is part of that too. And I consulted actually our counselor that instead of going for whatever number of genomes, why don't you select fewer, 100, and try to get not just the genome, but also proteomics data or metabolomics data from those individuals. So that is along your line of thinking. But on the other hand, I think we don't need actually to have the whole data space from one or few individuals. It's okay to have just more metadata to say, okay, we do have now 10,000 genomes from whoever, and we have 1,000 proteomics data from entirely different people. We are not that different in that sense. You know, the metadata will then stratify whether you are of a different, you know, I have to be careful how I say this now, you know, cultural background, if you want, right? And things like that. So I would be happy already with the attempt to go through those different data sets to different, I call them platforms, because these are different technology that provide then this type of data. But as I say, I see just these horizontal approaches right now where machine learning is used, but not the vertical. And this is, I think, something that will come eventually, yes.
Sure.
Great. So the syllabus for your class is available online. I just looked it up. So this would be a good thing for us to distribute to the cluster membership. So with that, oh, and the other thing I was going to say was particularly interested in your interest in 19264, the RFA. We too have an interest in it, and using radiomics and machine learning techniques to pull out from images response to immunotherapy is something that we've been working on. We had a paper in Science Advances earlier this year, and we'll circle back on that. So thanks very much, and I'd like to introduce our next speaker, Dr. Cesar Uribe, who's not yet at Rice, but will be shortly, and we greatly appreciate your joining us today. Cesar?
So thank you for the invitation. I'm really happy to share a little bit about myself today. So I'm going to be joining Rice, the EC department, as an assistant professor and the Louis Owen Jr. faculty chair. And right now I'm a postdoc at MIT. There is my email and my website. So some of the research that I do or the type of things that I do are listed there. So first is what I call decentralized machine learning. And I also don't like the AI term because it could be described from linear regression to convolutional neural networks and philosophy. So it's too much. So I work on decentralized machine learning, which is this particular case where you have multiple processing units working on a network. So this is sometimes referred as federated learning, which is like a hot topic these days in the machine learning community. And some applications that I work on or some of the things that I care about are things like, for example, computational optimal transport. So how can we design good algorithms for problems related to optimal transport? And in the next couple of the other slides, I'm going to talk a little bit about this. And let's say my main research thrust is in this idea of scalable algorithms, right? So, okay, if you cannot solve your problem with one computer, okay, you should use two computers, but how much harder is to coordinate two computers to solve this problem? So this is this slight balance of having more computational units to solve the problem does not always translate into faster algorithms. The coordination aspect appears now, and that is also a challenge there. So I work on finding ways to do this coordination efficiently, and I call this scalability. And some applications as well are on traditional problems on statistical inference, but then when you have like sensor of networks, for example, or in more recent applications could be like data is stored at different locations and you somehow want to apply processing to this data in a distributed manner. Some unique resources also, I was not too sure about what to put here. And first thing is this is a brand new lab that I'm opening. So tons of enthusiasm to work on new exciting things. I think that's one of the main things that I can bring to the table. I work mostly on analytical methodology. So I build good, strong hammers. Not necessarily work on the nails, but I work on having good hammers. And yes, and focus on methods and tools for large-scale data processing or decentralized data processing. And one of the unique resources is that I have a large international collaboration network. So I myself, I'm originally from Colombia. But then I studied in the Netherlands, and then from the Netherlands I moved to Illinois to do my PhD, and then from Illinois I moved to MIT, and now I'm joining Rice, but I'm also a visiting professor at MIPT, which is FISTech, Moscow Institute of Physics and Technology. So I have a large network of collaborators there, and then I work with people from Spain and from Hong Kong and Germany. So this is one of the things that I can definitely contribute to the cluster. So next slide. So yes, so what can I contribute? I mentioned this. So I have a bunch of hammers that I'm looking nails for. One of the things that I would really like to explore is how can we use this into medical data. Again, our strengths or in my lab is the decentralized architecture for distributed data processing, scalable methods. And yes, I mentioned this already. We have a bunch of hammers. And the main, let's say, motto of my lab is to change this idea from computation on the cloud to computation on the edge. And this is a recent trend in machine learning where we want to take advantage into processing abilities of local computational units. So some aspects is, for example, cell phones, right? How can we build upon this infrastructure where everybody has some form of computation and still guarantee some privacy and efficiency in their methods? And here I have a picture of all the, say, fields or tools that I work with. So I'm trained from optimization point of view. So that's my mathematical background, graph theory, stochastic analysis, and control theory. And of course, around there is a bunch of areas in which I have, let's say, touched a little bit, but not really expert in. So machine learning, medical data, sensor networks, and Internet of Things. So what type of expertise I'm looking or I'm seeking for the cluster? So I'm going to mention this in the next slide. So I really want to explore or want to find medical imaging expertise for some of the tools that I have developed of efficient algorithms, particularly related to optimal transport. And also field experience in medical data processing, because every paper on federated learning mentions that this can be applied for privacy preserving in medical data handling, but I'm sure none of those papers have really talked to somebody at a hospital to see how they really handle medical data. So I think this is a key issue that needs to be addressed, how to really build good models for what really happens in the field when handling medical data. And this is some of the things that I'm sure I can find in the cluster. And so since this is a brand new lab so far, I just have calls that I would like to apply for. I have received a small grants from companies like Yahoo or other small things, but like these are like the, let's say, house calls that I would like to apply, particularly the NSF size. And next slide. So in here, so I wanted to mention two specific problems that, let's say, I have like tools that I believe could be used for medical data, but I have no medical expertise or background to support them. So one of the problems that I work in optimal transport is this idea of Wasserstein body centers. And you can see here in the top left of this image, this number sevens plot. So here's a bunch of written number sevens. And if you compute, let's say, straightforward, naive mean of all these numbers, what you get is this blur of image because all the sevens have slightly different rotation, different scale, etc. But if you carefully design the metric in which you want to compute this average of the images, let's say you embed this data in one particular space, in particular the space of Wasserstein with Wasserstein metrics, you can actually recover this image on the right, which is a very nice representation of the number 7. So you can really extract the geometric properties of your data if you're careful about the metric in which you embed this. And when I was working on this, I thought, okay, imagine now that you have MRIs from the same patient that are slightly different versions of it because the patient moved or etc. And you really want to compute an average of these images. So the concept of computing carefully designed averages of images, in particular medical images, is something that I'm interested in. And again, this is just an idea that I think might be interesting. Of course, if somebody works on actual MRIs, might tell me that it has no reason to do it this way. But and then the other image on the top right, what I have there is these images from cells. These are cells that are from people working at the Broad Institute here at MIT. And what they really want to understand is how the morphology of the cells changes with different gene therapies. So again, the question is, how can we compute the average shape of cells, normal cells versus different cells with different therapies? So it seems like optimal transport and the ability to compute average of different objects, in this case shapes, might be of interest for medical applications. But again, I have the tools. I don't have the actual data or medical collaborators to explore these ideas. And in the bottom, this is just, again, these ideas, let's say, they call this in the news every time. So one drone is not enough, then use 2 drones or a swarm of them. But then when you do this change, you introduce an additional layer of coordination that you need to take care of. And so this is not as straightforward. And this relates to this idea of federated learning that is being used recently in a bunch of times. And I think that it needs to be approached carefully, especially when you want to apply this to medical data processing. And yeah, this is all that I have for this presentation.
Thank you very much, Cesar. Any questions? So a comment I wanted to make was, yeah, absolutely. There are plenty of people in this cluster who understand medical imaging and the intricacies of medical imaging quite well. And personally, I would love to hear more about the approaches you have in mind for normalizing images. I wouldn't say there are a lot of applications, but there are definitely some applications where that might come in interesting. And there are actually, on the cell image side, there are a number of metrics that we already use for shape, but we can always use more. So that would be interesting as well. Other questions?
Yeah. So this is particularly when I was doing this experiment on this form of MRI data. What I have is I couldn't find 4 images of the same patient at different locations. So what I just run is I'm computing the average of 4 different patients, which has it makes no sense computing average of 4 patients, but just like let's say experimental mental experiment just to see what happens. And I think this is
on the MRI side, there's actually a couple immediate projects that I would love to talk to you more about. There's one on a pediatric cerebrovascular disease called Moya disease that we've been studying for a while, where the vascular morphology is modified as a result of the disease. And we've quantified it, and that's been published. But to find ways of normalizing the morphology in order to come up with standard geometries to do, for example, fluid dynamics calculations on, this would be really, really interesting. So we'll certainly talk about that some more. Thank you again.
Yeah, of course. Thank you.
With that, I'm going to hand it over to Dr. Devika Subramaniam, who will handle the next section of this meeting.
Very good. Well, we have Sebastian Acosta, who's going to introduce himself from Baylor College of Medicine. Take it away, Sebastian.
Sebastian, you're on mute.
Can you hear me now?
Yes.
Thank you so much for the opportunity to introduce myself to this group. I'm an assistant professor at Baylor and at Texas Children's Hospital. By training, I am a mechanical engineer and an applied mathematician. I got my PhD from Rice not too long ago, and I found a job just across the street, so I didn't have to move from Houston. The type of work that I do at Baylor and TCH is on machine learning and the analysis of data from patients. One of the main things that I am interested in is the integration of mechanistic models of the cardiovascular system with machine learning methods to monitor patients in the ICU, the intensive care units. As you know, in these units, patients experience life-threatening events, and it's hard for humans to anticipate these events in advance. And this is the case for doctors and nurses who work there. By the time imminent symptoms appear, it's a little too late, and the care for these patients become more reactive rather than proactive. So this research focuses on developing the artificial intelligence methods to be able to predict these events in advance. Another area of research is on photoacoustic imaging and ultrasound imaging, and also, again, integration with the physical understanding of how these methods work with machine learning to improve resolution and quality and overcome some challenges for these imaging modalities. One of the unique resources that we have at TCH is called the Sickbay Database. And some of you may be familiar with this system. And basically, it's a very well organized database where we have time synchronized records of the vital signs that the patients produce and their transfers within the hospital, their lab results, and the medications administered to them. The next slide. What I can contribute is my expertise in biomechanics and numerical methods for dynamical systems and partial differential equations. Also on the analysis and the construction of methods for inverse problems and imaging and control theory. Also on signal processing is a big part of our work, since we have to process the waveforms and the vital signs produced by the patients. And in general, the methods of machine learning. What I'm seeking from the cluster is precisely modern methods of machine learning, like deep learning, data mining, anomaly detection, and classification methods that can work for this very complicated, sets of data, and also in a distributed manner, since we have massive amounts of data coming from different computational units. And of course, we're also interested in funding from the NIH, the NSF, and the American Heart Association. The next slide. Oh, I had a third slide. It's okay.
I only have the two. I'm sorry.
No, it's fine. So basically, I had some pictures about these problems of, to give you a better idea what Sickbay was, or what Sickbay is. The amount of data that we have available is very massive, and is very rich in how we can monitor these patients. And again, back to this idea that we can use, our goal is to use both the physiologic understanding of the patients, like the cardiovascular system, the blood flow, and the mechanics of the heart, and so on, and integrate that with machine learning approaches to be able to infer the state of the patient in advance, before to be able to give doctors and nurses some time for them to prepare for certain changes that the patients are experiencing. And that's it.
Okay, thank you, Sebastian. We have time for 1 or 2 questions.
Yeah, hey Sebastian, this is Craig. Could you tell the group? Hey, could you tell the group exactly how much data we have access to? Because I think it's a little bit different than it's a little bit more than massive.
Yes. So I have, this is what I had in the third slide. The system started collecting data from patients at TCH in January of 2013. We have, I believe, more than 300 beds connected to the system, and we record data 24/7, every day of the year, every time there's a patient in that bed. Using that monitor, the database is collecting that data. It's more than 1,000, what we call bed years of collected data, more than 1 million patients that have been admitted to the hospital since then, more than 100 million lab results, and more than 70 million medication administrations. So we have this rich interconnected set of data that characterizes the state of the patients, and this is what we get to play with.
Yeah, I had a similar question. So now we learned how deep it is, so million N's, right? What about P? How many features, roughly?
Well, the number of simple vital signs, it's about, how many do we get usually? Like 15 or 20 different vital signs. Some of them are more comprehensive. They have the actual waveforms, like the electrocardiogram, the arterial blood pressure, and so on. And in terms of the number of labs, I don't know, it's in the hundreds of different type of labs, and also medications.
Could you talk a little bit about how you set up that data set? Might have been set up before you joined, but I think that's important. As working on the clinical side, we do have a lot of data, but harnessing it and making it accessible for analysts is a challenge. Yeah.
Well, I think Craig is better suited to respond to that. He did it.
Yeah, so you're absolutely right. So I wrote Sickbay, and so we spun it off to a company called Medical Informatics Corp that's commercializing it. But there's an entire standard development kit and research API that allows you to both ingest data and as well as kind of do the research process, which generally for us has kind of 4 steps of rapid algorithmic prototyping, large-scale retrospective analysis, so distributed computing without necessarily having to, write distributed computing code, distributed ML using Spark, and then a large-scale prospective real-time evaluation. And so we have an environment that allows programmers to go and develop all of this code and then deploy it effectively in production. Obviously, production for research purposes, but it can even be, then taken through the FDA and put into clinical practice if necessary. So there's a whole bunch of different methods and tool sets that we have to take this data and do stuff with it. I can get more specific if you want, but I would love to show you if you're interested.
Yeah, maybe I'll just contact you. If you can text me on chat your email address, I can reach out to you.
Yeah, I can certainly do that.
Thank you.
Any other questions?
Yeah, the Sickbay system is definitely one that is worth following up on. I was actually not aware of it until Craig and Sebastian introduced me to it in the context of a grant we were writing a couple months ago, and the capabilities are just awesome.
Well, if we have less than 15 minutes, or 15 minutes, so let me invite Dr. Rumasa Chana to, I hope I got your name right, tell me if it's wrong, to present here in our little consortium. So,
thank you, Devika. Thanks for the introduction. So I'm a clinician. Shifting gears from the previous talks, I am a retina specialist and I see patients with diabetic retinopathy day in and day out. And what I'm really interested in is how to apply all these wonderful innovations related to autonomous algorithms and AI in the clinical world to make clinical care better and to make discoveries. And so, if I hone down on my research interest, it's implementation of these innovations in clinical care and using these advanced data analytical techniques that you all talked about, especially Dr. Lance touched upon some of them, to understand pathways of vision loss. So we've done a lot of studies to understand why people with diabetic retinopathy lose vision, but a lot of it has just been based on, the more superficial demographic, basic retinal imaging data that we have, but we don't really have studies that have looked at the depth of data, so the genomics, the metabolomics, the proteomics, and put all of that together to figure out why some people lose vision, why some people just have mild disease, while some people go on to develop blindness. So those are kind of my research interests. The resources that I bring in is my expertise in retinal disease. We also have a retinal image reading center, which is considered the gold standard for interpreting retinal images. So before any, so in diabetic retinopathy screening, we're actually unique in the sense that we have an autonomous algorithm that can detect diabetic retinopathy and also refer a patient for further evaluation to a specialist without needing a doctor at any point in that process. But in order to get there, it was decades worth of developing the algorithm, and then it was also bringing it to the clinical practice. And in order to do that, those outputs had to be compared with gold standards. And so the reading center is kind of the gold standard, considered the gold standard right now, against which this AI was compared. And then the other thing we have is a robust clinical eye research unit as part of our shared resource, which has lots of coordinators, lots of imaging, basically any data that you would like to collect on the patient, we're able to do that. So it's really helpful when conducting human subjects research to have that kind of resource available. Next slide, please. So my expertise in diabetic retinopathy, that's been my area of research, human subjects research, retinal imaging. In implementing AI, we just did a study in which we implemented AI, which is already approved by FDA for use in adults. We implemented it for diabetic retinopathy screening in children, and we were able to show improved compliance with diabetic retinopathy exams from 49% to 95%, because basically it was right there and everybody agreed to get the AI and were able to pick up more cases of diabetic retinopathy. The expertise I'm looking for is deep learning expertise to use all these new algorithms to healthcare data, to mine healthcare data, and use retinal images to extract more data than what we currently extract from retinal images. And similar to what I talked before, the RFA, this is an RFA that we're currently working on, which basically looks at how we can use these innovations to address the impact of COVID-19. So with COVID-19, a lot of normal or chronic diseases patients are kind of not coming to the clinic because they don't have until they have an acute problem. And so how to utilize these innovations to provide healthcare to them while minimizing their contact with physicians. So that's kind of what the RFA is centered on. And we're planning a randomized control trial, randomizing patients to an AI intervention versus standard of care. Next slide, please. So this summarizes some stats about diabetic retinopathy and some of our recent papers. One part is implementing it in the healthcare workflow. Doctors really worry about, well, if I add this step, is it going to delay patient care? Is it going to increase my time per patient? Will I be able to get through my clinic? And then what policymakers and hospital admin wants to know is the cost effectiveness. So if we're going to change our care or bring in a new solution to a problem, how cost effective that's going to be. So that's another aspect that we look at is cost effectiveness of these interventions. Thank you. Thanks for having me here, and look forward to talking to more of you about this.
Thank you. So the diabetic retinopathy, is this, have you worked with the Google team in helping on, whoa.
Yes, so I was working with the Google team initially. That was several years back when they developed their algorithm.
Right.
And then the algorithm that we tested in the clinic was from the Iowa team that got FDA approval for their device. So they're the only ones right now. There's another one, iNook, that's got an FDA approval.
Okay.
So I've worked a little bit with all of them.
Oh, terrific. So yeah, this is I think this cluster, I mean, I hope you find, I mean, I think you have lots of choices of collaborators here. A lot of us who have expertise in deep learning who would love to help you, and of course the kids and the adults that you serve. You know, eyes are the most important thing we have. Well, everything is important, I guess, but eyes definitely. And if you can intervene early and take care of people and, you know, and detect disease at a much earlier stage, I can see the advantages of that. So, and the fact that you're ready to run a clinical trial on these things, I think makes it really, really cool. So, you know, count me in as someone excited. So
Thanks, Devika.
yeah, sure. But I'm sure I'm not alone. I mean, you'll have tons of people interested, which is good.
No, this is
Sorry,
great.
we lost you to Wisconsin,
But she can still be a remote member, no? I'm hoping.
That's the fun part about COVID.
Yeah, she's in our backyard, and yet she's
every cloud has a silver lining.
Yeah, I guess. More questions for Dr. Chana.
If not, maybe I can close the meeting, Devika, if you.
Okay, sure. I mean, but if there are questions, why, you know, yeah, then go ahead. Looks like.
So if there are any questions for Dr. Chana, I would like to take the opportunity to thank all the speakers for the very exciting talks. As Devika mentioned, we have a lot of people with hammers that can help the people that have nails. And I think Dr. Lance asked, what is this cluster all about? So let me take a stab of what I think the cluster is all about. It's about bringing people together, bringing people that have hammers, bringing people that have nails, and working together. And at least as far as I am concerned, you don't have to work in a Gulf Coast Consortium institution. I know the rules say differently, but you know, we all are scientists here, and we enjoy working with interesting people because we don't only bring our brains to work. So one of the things I would like to point out that it's not only the people that are here, you know, the membership is growing, and we have other clusters, and the leadership is making conscious efforts to reach out to the other clusters, appoint liaisons. So we hope to be able to be your matchmakers, if you will. You know, as Anat mentioned, oh, I can see this application, maybe she can connect some people, so on and so forth. So feel free to send us ideas on how you want this cluster to evolve. I mean, this is a grassroots effort, not a top-bottom effort. So feel free to email to all of us or any of us on how we can make the cluster serve you. Because I'm very thankful that you took 1 hour from your busy schedule to attend. And I really like that you guys proposed specific calls that you are interested. I'm sure you'll get a lot of contacts for something like that. And we want to promote creating collaborations, tight-knit collaborations that go even beyond a single grant, but are evolving in terms of bringing people together and working together, both in research and education. I mean, Dr. Lance mentioned some educational activities. The cluster wants to promote education. So please send us your ideas for this, or how can we support you, how we can be the matchmakers, so on and so forth. So I don't know, Devika, do you have something
Yeah,
to
well,
add?
I just wanted to say that education will be a key part of what we're trying to do. Already there is a small group, Rumasa is part of that group. And already there are, today I learned something new, a new course from Dr. Lanz out here. So I think we can try to put together all the resources we already have in the form of courses we teach already. So maybe I'll reach out to you, Rumasa, because I'll be liaison from this group, but then we need someone to kind of lead that effort. And so publicly here, I'm asking, please serve in that capacity. And we have the mailing list. Everyone's invited to be part of this group. This is not an exclusive group. So we'll try to gather the names of, so reach out to either me or to Rumasa, and we will get you into this mailing list, and we will start thinking about how we can have small workshops. There's a lot of interest, it seems, on graph convolutional networks. We can have a workshop on that. Maybe one on, topics of different interest to us, generative models, deep learning, how to set up a deep learning pipeline, something basic, something more advanced, the whole range, I think, to accommodate our membership and to accommodate the different range of problems that we are going to be solving here. So yeah, as to just repeat what Yanni said, really, this is about bringing, people with hammers and, or actually what Carlos said, that's where it started, with people with problems, and hopefully together we'll be stronger. That's the goal.
So another thing that, it's happening, as you said, Devika, we are forming subgroups, people that are interested in a specific call for proposals, people that are interested in education, people that are interested in palliative care, for example. Yeah. So we're trying to energize this and support these groups. So I would like to invite all of you to reach out to your institutions and invite people to just join us the next time. The next event will be in January 11, 1 to 2 p.m. And please volunteer to speak. Even if you don't have these 3 slides, talk. Let us hear what you want to say. I mean, we want to get to know you. If you don't like the slides, just talk freeform, okay? It doesn't take a lot of preparation, but we want to hear from you. So please volunteer to talk. Please encourage people. There is no formal applications. Just forward the email, they sign up, and you get into the mailing list of the cluster. And that said, I want to announce that the video, the recording will be available in the website of the cluster. Unfortunately, we're running a little bit behind, but eventually it will be. So we'll notify everyone on this merged list that this is available. So those of you who spoke today, you didn't have only 36 participants, but probably will have 136 views as we move forward.
Yes.
So with that said, I want to thank you. Wish you a happy Thanksgiving, happy holidays, and we'll see you back in January, if not earlier, some of you. So thank you very much. We appreciate it. All the thanks go to you, not to us. Thank you, everyone.